Hello everyone and a warm welcome to today's session. My name is Edwin van Vulpen, I'm the head of clinical BD in Europe and I'm the moderator of this session today. Today's session is all about a comprehensive overview and practical guide of a human radiolabeled mass balance studies for regulatory submission. Our speaker, whom I will introduce in a minute, will provide a comprehensive overview and practical guide to get the most out of your human radiolabeled mass balance study, covering the many nuanced steps and considerations. Our speaker will walk through the main objectives of a human radiolabeled mass balance study and the ethical and regulatory consideration. In addition, he will cover formulation of mock batches, releasing of a subject based on radioactivity, an overview of the timelines, data analysis and reporting. To better describe the various study types and components, our speaker will present 3 case studies based on recent experiences in our QPS Netherlands facility. QPS can offer all services required for a traditional human radiolabeled mass balance study, including studies with radiolabeled compounds and microtracer studies. Let me take you through some of the house rules. The webinar is being recorded and you will get an email after the webinar with a link to watch the recording on demand. Submit questions during the webinar using the question box. As many questions as possible will be answered during the Q&A session after the presentation. If a question isn't answered live, we will follow up with an answer in an email after the webinar. Now, let me introduce you to the speaker, Wim Taminga, Senior Vice President, Global Head of Early Phase Clinical and Site Head of QPS Netherlands. Wim joined QPS in 2010, and in his position, Wim oversees QPS's fast-growing early phase clinical business, Phase 0, Phase 1 and 2A, in the US, in Europe and Asia. In this role, Wim leads a large team of cross-functional clinical development personnel, and under his leadership, more than 100 human radiolabeled mass balance studies have been performed. Wim received his PhD in pharmacology, pharmacogenetics from the University of Groningen in the Netherlands. Wim, please go ahead.
Good day. Welcome to the first part of the QPS webinar on mass balance studies. Today I focus on the clinical execution, and part 2 will be on bioanalysis and metabolite profiling. The agenda. I start with an overview of how to conduct a mass balance study. We discuss important questions like normal radioactivity levels or use microtracer levels. Can studies be conducted in healthy volunteers? Should you collect samples for pharmacokinetic and analysis on these studies? We end with 3 case studies, all with specific elements that may help you understand where to think of in planning these studies. So, how to conduct human hAME trials, which I call mass balance studies. Mass balance studies are executed mainly to answer questions on the fate of a drug when entering the body. How much ends up where, which metabolites are formed, and how and where is it eliminated. There is no clear guidance on how to do it, but guidelines tell why. The FDA guidance provides recommendations to identify drug metabolites whose toxicity needs to be evaluated. They encourage identifying any differences in drug metabolism between animals used in non-clinical safety assessments and humans. The EMA guideline on the investigation of drug interaction says that mass balance studies provide information on systemic metabolite exposure as well as data used to estimate the main elimination pathways. In Appendix 5 of this guidance, some principles on the how to execute a mass balance study are given. We have 2 types of radiolabeled studies. By the way, when I say radiolabeled, I mean that the IMP is labeled with a radioactive compound, usually carbon-14. So two types of studies: microdose studies, low radioactivity dose, either with normal IMP dose or low IMP dose. Traditional mass balance studies, where radioactivity levels of 100 microcurie are given. Because of low radioactivity levels in microdose studies, you need a more complex methodology called accelerated mass spectrometry. You can give radioactivity to healthy volunteers, but you need to assess what the risk is for a human when they get radioactivity. This is called dosimetry. So a certain amount of radioactivity represents a risk to an individual, which is calculated in millisieverts or rem. It is generally accepted to dose radioactivity up to radiation burden levels below 1 millisievert or 0.1 rem, which is less than the normal annual background dose we get. Higher doses can be considered, but certainly need a better justification. The maximum dose is normally around 100 microcurie. This is normally sufficient for the required measurements. As we discussed earlier, metabolism is always the main objective of a mass balance study. Another important element is to assess the mass balance. Did we catch everything that we dosed? This is normally greater than 80%, and the more you miss, the more questions you can expect. The route of excretion is an important parameter to understand PK and DDI interaction. This is also the case for the total radioactivity profiles in plasma and whole blood. Keep in mind that the labeling of the drug is really helping to get this data. Without radioactivity, these objectives is much more difficult to achieve, if not impossible at all. Timing of a mass balance study is debated, but most companies do it when starting to complete their NDA filing, normally between phase 2 and phase 3. As a mass balance study needs to be planned ahead, the whole process easily takes 12 to 18 months. In the Netherlands, there is no radiation board that is involved in the review process. Many other countries in Europe have these. For example, in England, you have the RSAC. And this means that besides some additional documents, the regulatory process is the same as in any other CTA. In general, CTA application in the Netherlands is fast and efficient. We have a parallel submission where documents are submitted to both the competent authority as well as to the IRB. The IRB is also responsible for the design and the scientific review. This leads to fast CTA approval, normally in 2 to 3 weeks. For a mass balance study, dosimetry calculation is required. This is normally based on QWA studies. Quantitative whole body autoradiography is an in vivo study in which rats are dosed with radiolabeled IMP, and at successive time points, radioactivity is measured in cross-section slides of the whole animal to show distribution over time. Chemistry, manufacturing, and controls for radiolabeled compounds is one of the most important aspects. Thorough preparation is required. This is especially the case for the position of the radiolabel and the used isotope. Preference is to use carbon-14 and build this isotope in the IMP at a position that is not lost in an expected metabolite. Also, it is important to consider how your compound will be dosed and whether the compound has the right properties. The vast majority of mass balance studies use an oral drinking solution. But if the compound is not soluble in the drinking solution, then this creates a problem. We had many cases of this, and additional efforts were put in creating a stable suspension or something like that. In addition, adhesion to dosing material can create issues, especially when IV dosing. Shipping radioactive material requires solid planning and preparation. You need an export license and an import license. This requires a few weeks. And with most of these processes, many details are required in applying, and missing a small detail will hold up your expensive material for days. Keep in mind, a good start is half the work. Once the compound is received, the dispensing radiopharmacy needs to release the dosing solution for dosing. This requires initial qualification of the methods that will be used for dosing. A mock preparation is conducted to prove that things are working fine. This whole process easily takes 2 to 3 months of preparation, which is shown in this detailed table. Not all CROs have these capabilities, and therefore other vendors have to be involved as well. This complicates the process. QPS can conduct all these steps. In running a mass balance study, a site also needs the equipment to do the analysis. This is of essence as most of the analysis is time critical. For example, before subjects are sent home after being in the clinic for 5 to 10 days, they are required to be checked on their radioactivity levels. Not having access to the machine would slow down this process significantly. A liquid scintillation analyzer is a machine that can measure carbon-14. A radio flow detector is a machine that can measure carbon-14 online in an HPLC setting. An oxidizer is a machine that can combust feces and/or whole blood in order to count total radioactivity. At QPS, we have special labs with all of these machines, as well as the radio license to execute this work. Once samples are collected for a mass balance study, the first step is to measure the total radioactivity. Just measure how much radioactivity is present in a sample of a certain matrix, for example, feces, plasma, or urine. Keep in mind that this still doesn't require any separation of metabolites. You simply measure the total amount of radioactivity present. In part 2 of this webinar, Dr. Fred van Heuvel will explain many more details on the analysis. It is also important that you collect all materials that may hold radioactivity and will contribute to the mass balance. For example, the empty bottle of a dosing solution may still contain 1% of the actual dose, and that should be used to assess the total mass balance. Another example is if a subject vomits within a few hours post-dose, and this will contain radioactivity that you need to complete mass balance. In some cases, expired air is a route of excretion, and therefore you also need to collect samples of exhaled air. In the next few slides, we cover a few specific topics. The first is human ADME study or a microdose study. So looking at a microdose study, the pros are there is a minimal radiation exposure. They can be executed in any unit. Actually, I need to say in any unit except for a unit that does a lot of normal mass balance studies because that may contaminate your unit. No radiation burden calculation is required for the IRB. There is no need for GMP status of labeled compounds, so not for the unlabeled compound, but not for the labeled compound. And this is an easy way to assess absolute bioavailability, and there are no specific requirements for laboratories. On the contrary, hAME analysis is complex and expensive. hAME is the technique that you need to measure the samples. hAME is also more time consuming than liquid scintillation counting. For a traditional mass balance, so about 100 microcurie, the pros are that liquid scintillation counting is cost efficient and it is a fast methodology. Turnaround times are fast. Online liquid scintillation counting is possible. The amount of radioactivity improves the sensitivity of the analytical and METAP ID activities. On the contrary, radiation burden calculation is required and requires a dosimetry report. Amount of labeled compounds to be manufactured should be GMP or GMP-like, and collected samples are considered to be radioactive. The preference is to run a human mass balance study in healthy volunteers. This saves time and money in recruiting patients. With oncolytics, this is difficult. However, if an oncolytic is non-cytotoxic in most cases, these studies can be conducted in healthy volunteers. If required, human mass balance study can be conducted in oncology patients. QPS has, for example, a network with 2 oncology centers that will refer patients. So it's a small question, but still an important question is, do we need to collect samples for normal PK measurements? And in many cases, this is in the protocol. QPS is of the opinion that to measure drug levels by, for example, LC-MS is normally not required. These samples have been measured in many other studies, and the overall sample size, normally between 6 and 8 volunteers, doesn't add a lot. If you still want that, you need to find a lab that is capable of running radioactivity samples in their LC-MS lab. So the next few slides are on a couple of case studies. And the first case study is an example of an oral drinking solution. In this study, we dosed 14C labeled PBI 4050, and this study is published, after a single oral dose of 1200 milligrams to healthy male subjects. Those subjects aged 21 to 55 years. We collected blood, urine, and fecal samples. And so during the first 48 hours, we did it with regular intervals, and later on we did it with 24 hours intervals. And the subjects were discharged from the clinic no sooner than day 6, and we checked whether the radioactivity levels were following one of the criteria. Either the plasma whole blood radioactivity levels were below the limit of quantitation in 2 consecutive samples, and the total excreted radioactivity is higher than 90% of the administered dose, criteria 1, or criteria 2, 2 consecutive urine samples are below 1% of the administered dose, and 2 consecutive fecal collections intervals are below 1% of the administered dose. Safety and tolerability were measured. It's of course not one of the primary objectives, so it's normally fairly standard. AEs, vital signs, ECGs, and clinical safety labs. This shows the results of the study. The overall recovery was 87.3% in 5 days, of which the by far majority of the excretion was in urine. A very minimal amount of drug was excreted in the fecal samples. The drug was absorbed quickly and also eliminated pretty quickly, which you can see in this picture. And also, from these results, we could conclude that there was no accumulation of the 14Ch labeled compound in whole blood, in red blood cells. After the study, we did conduct the METAB ID, and we discovered a few unknown metabolites, as well as we could also clearly detect the main metabolite, the M221 over here, and the other main metabolite, M333. So this is done after, of course, all the other samples have been analyzed. So in conclusion, for this drug, mainly eliminated in urine, the cumulative recovery in feces was very low. There was little or no distribution to red blood cells. And then we studied the major metabolic pathways, where overall, and we could also assess how much of the drug, the unchanged drug, was excreted in the urine. And overall, the safety was well tolerated. But I cannot even remember one study where tolerability was an issue in a mass balance study. So the next example is a case study of a single intravenous dose, and this also holds another specialty, and which I will explain in a minute. So in this study, we dosed a single IV dose to healthy volunteers. Once again, the objectives are metabolite ID, where mass balance, and profiles in plasma and whole blood. Again, we dosed, and you can see kind of the same with the previous study, 22 to 58 years old. So it's kind of our standard volunteers that are willing to do that. For a specific reason, we co-administered clemastine, which was a specific requirement for this study. We collected blood, urine, and fecal samples. And then the same with the previous study, we collected in 24 hours intervals after 48 hours. And in this case, subjects stayed in-house until day 9. And of course, they could leave the clinic earlier if the discharge criteria would be met. It should be noted that in this case, it is not a drug, but it is a imaging agent that is used in patients with tumors that overexpress certain receptors. So in this case, it is expected to behave different than a normal drug, and which can be seen in this this specific picture, where you can see that the overall recovery is low. Actually, we found an average recovery of 17% after in a period of 9 days. And after day 9, subjects were dismissed from the clinic and were asked to collect, and because there was still a lot of radioactivity present, they were asked to collect urine and feces samples. And those samples were brought back, and not all, of course, but at certain time points they were asked to collect samples, and those samples were brought back to the clinic at some study visits and a follow-up visit. And after analyzing these samples, interpolation and incorporation of this additional excreta resulted in an estimated total recovery of 35%, which is actually pretty well in line with what was seen in preclinical data. The METAP ID was in this case not really very specific, but we did do that, and we kind of confirmed what the client was expecting. The final study that I want to show is a case study where we dosed via nasal spray, and this study also holds very specific things. So the objective in this case was to investigate the absorption, distribution, and metabolism of a compound that is to be administered via a nasal spray with a single dose in healthy male subjects. 6 subjects in this case, 18 to 65, and then once again intranasal application. Blood, urine, and fecal samples were collected, and then again the same, 48 hours with regular intervals, and then later on larger intervals. Concerning the nasal application, this was very specific. The device was specific, and we want to make sure that things were dosed properly. So we had on day -1, we did dedicated training of the dosing personnel as well as the subjects. In dosing, 100 microliter was administered via spray in each nostril. People were not allowed to blow their nose, were of course preferably not allowed to sneeze, but that's not something you can control, for at least 10 minutes after dosing. And we allowed to use wipes during dosing and thereafter, and those wipes were were counted for radioactivity. And with this client, we had multiple discussions about kind of occlude the nose, so dose and then kind of block it by tampons or whatever material that could block it. But because of the kind of occlusion, they were afraid that that would affect the absorption of the drug, and for that reason we decided not to occlude anything and just leave it open and use wipes. So the study is currently under full analysis, but these are the initial results. And the first thing you can see over here is that the excretion, the recovery, is really very variable. So some of the subjects have a recovery of about 80%, and some subjects only have 40% recovery, which of course is not what you want. So besides the urine and the fecal samples that contained the radioactivity, in this case the majority was I think it was about 50-50 fecal and urine samples, but the majority was in the tissues. So the green bar shows what was in the kind of the initial tissues, and then the later tissues also still contained, so after the 30 minutes still also contained radioactivity levels. And this shows how complicated it is and how important it is to collect all kinds of details during collect wipes, collect cotton swabs that you use, whatever, to clean things, and measure those as well to assess your total radioactivity. So in conclusion, human ADME study requires thorough preparation with a focus on detail, the right radiolabeling, careful consideration of the right dose of radioactivity, not too low, but also not too high, the right route of administration, and sufficient sample collection time. Use the right expertise in designing a human ADME study. And this is my experience over many years. A solid design pays off. So rather spend 2 additional months in planning for a study. So plan well ahead instead of rushing through a study and then at the end have incomplete recovery. And it should be multiple people involved, so clin farm people, pharmacy people, lab people, and also MedTap ID experts, which are kind of at the end of the process, but it's very important that they also provide the team with input on where the radiolabel is and things like that. Most human ADME studies can be conducted in healthy volunteers. If radiation burden is not rate limiting or a normal human ADME study is faster and more cost efficient, oral dosing is the easiest way, and if possible, this is preferred. Other routes of administration are possible, but will complicate the study conduct considerably. And at the end of the day, I would say work with a CRO that have the experience and can conduct all elements of a study. And with that, I conclude my presentation and thank you for your attention.
Thanks, Wim. Let me remind the audience that the question box is open, so don't hesitate to raise any questions that you might have. There are already some questions. First question, Wim, for those opting for microdosing, do they also tend to go for non-GMP preparation?
Yes, but in many cases this is possible, as for doses of 0.5 to 1 microcurie, the amount of API applied as the percentage is below 1.15% in several studies. And if it's below 1.15%, the API may be considered as an impurity instead of as a formal GMP preparation. So yes.
Okay, thanks. Then somebody is asking how to be sure that we have linear dose exposure response when we use microdosing to assess bioavailability.
Yeah, so the question should be more like you should be aware when you use microdosing studies that in previous PK studies you have collected that kind of information. So it is not that it is something that you will get from the study. No, you need to know that up front. And if you know that, then a microdose study is okay. If you don't know that, then it may be very tricky because you may draw conclusions that are would be based on a different profile at a very low level.
Thanks. Then somebody in the audience is asking what extra considerations are there for study design for an extended release formulation with long half-life?
Yeah, so in general, so that shows the second example. You need to prep. Number one, you need to prepare your subjects to be able and to be willing to be in the study for sometimes many months, and that can last up to 5-6 months. That's number one. Number two, you need to have a very careful scheme to assess where you are when subjects leave the clinic. So if your fecal samples and your urine samples are still pretty high, so above the 1% per sample, which is or maybe above 2.5% per sample, something like that, then if possible, it is preferred to keep them in-house because collection at home bears a certain amount of risk. So you can do that. And the other thing I think which is also important is to consider that after a while, the technique that you apply to measure, for example, standard liquid scintillation counter, may not be sufficient enough to measure the radioactivity level. So you maybe need to go to a technique that can measure lower counts. Because at the end of the day, if you collect urine for, I would say 10 days, that's a lot of urine. And of course, I'm not saying that you have to collect it in one batch and bring it to the clinic, but it means that overall the concentration is very low. The total radioactivity levels are very low. So that's the third element. You need to be sure that at the end of the day, you can rely on more sensitive techniques. And one thing that people could say, okay, so you can use then hAME for that, for these type of samples. And that may be tricky too, because at the end of the day, hAME requires very low levels. So sometimes even in low levels that are not easy to measure with liquid scintillation counting, the levels are still too high for hAME. So those are the considerations. So volunteers, have them be willing to stay in-house for a significant period of time, I would say 14 days, then have them be willing to collect the samples for a longer period and have sensitive analysis techniques to measure it. Otherwise, your study can be very difficult to assess your mass balance.
Okay. Next question. Would you recommend to perform a microtracer absolute bioavailability within a human hAME study? And if yes, in which cases?
Yeah, so that's kind of difficult question. So I would say that there are different advantages. So both parts can in principle be done in the same subjects. So there is no need to have fresh subjects. If you microdose to assess the bioavailability and you do a normal radioactivity dose, that still can be done in the same subject. And this is especially handy when you have complicated subject recruitment, for example, oncology patients or things like that. When that's not the case, I don't know, it's really worth the investment and just do it in a parallel design where you dose or use fresh subjects to do it, because at the end of the day, subjects need to be willing to stay longer in the clinic. It's a crossover design. People maybe are maybe not willing to stay and things like that. So for healthy volunteers, I would recommend, yeah, it really doesn't add a lot of value. When it's difficult for whatever reason, technique or patience, then I recommend that you do it that way.
Okay. Then somebody's asking, are you measuring urine pH routinely before analysis?
No, we don't do that unless there is an indication that there is an instability in the samples or in the urine samples or something like that. But normally we just simply measure the just measure the total radioactivity. I'm not sure, 100% sure if the question relates to instability, specific things. Of course, we can do that, and I would recommend you to also listen to our second webinar where our analytical experts will talk. That webinar is scheduled for, if I'm not wrong, Edwin, correct me if I'm wrong, on June the 16th, where they will talk about analysis, METAP ID, very specific, very detailed, where you maybe should kind of rephrase that question and ask them what they think. But for the total radioactivity levels, we normally don't do that.
So that same person is responding directly, and the question is, having the urine pH for ionized compounds like memantine can give you detail on the PK.
Okay. So that is certainly a question that should be asked in the next audience, if that person is willing to do that, or shoot us an email and we can comment on that, how we would do that from a MATEP ID and analysis stability perspective.
Yeah, we definitely will follow up. Okay, great. Hey Wim, what about CSF levels? Are they measured as well?
So normally I would say in the Netherlands, so we have multiple clinics, but the Netherlands clinic is proud to do the most complex studies in the world. At least I'm proud to do those studies. My operational people are not always that proud. Once they finished it, they are proud. But CSF in a mass balance study, I never done that. We did a lot of CSF studies, a lot of continuous CSF studies, but a combination with total radioactivity, we've never done that. But I would be certainly open to discuss that with somebody.
Okay. Yeah, and we already come at the last question. This question is related to the missed guidance. So the missed guidance refers to steady state metabolite percentage, assessed versus 10% of total. How do you make a connection between a single dose ADME study and steady state exposure?
Yeah, so that is a kind of the same question we discussed before. That is information that you need to know upfront. So we have done studies where we did a loading dose with a cold compound for a few days to reach steady state, and then do on top of that total radioactivity, a radioactive level. And it is normally known upfront how important that is. So if that is required, cold pre-dosing until you reach steady state, and then do a total radioactivity, a radioactive dose on top of that.
Excellent. Thanks. So if there are no further questions, then it looks like that we're gonna close earlier. I would like to thank Wim for his presentation. I would like to thank the audience. I would like to wish everybody a good day, a nice day. And please remember, we'd love to hear from you. So if you have any questions, remaining questions or suggestions, then please let us know. And then for today, thank you very much. And hopefully we'll see you at our next webinar. Thank you and bye-bye.